Hello and welcome back to Head of the Epi Curve. This is the first video in a new series that we're going to be starting on computational biology research, pivoting away from previous public health videos that were posted on the channel. Those will still come out, but for a while now we're going to be focusing on some computational biology research tools. On today's video, we're going to focus on a program called GEO from NCBI, and it's a very good tool for finding online data sets about genetic research. It's a tool that I've used multiple times in order to form the basis of my computational biology research. So to start off, I have a bookmark here. It's GEO, but you can find it just by looking up NCBI GEO. And it's the Gene Expression Omnibus. This is the link. Now, obviously, this screen's a little bit different than the one I have bookmarked, but they're both the same. And so we can look up a data set for a specific disease. Say you want to do research into schizophrenia. I've done research into schizophrenia. Type in schizophrenia. Look at all these different things here. You've got all these results here. But I'm going to focus in on a very specific type, so I'm going to go back to the link that I've got up here. This is specifically the microarray area. We go schizophrenia here. And you see how this gives me all these entry types and these specific study types. This is what we want. And so the easiest way to be able to do your research is to make sure it's an expression profiling by array. This will allow you to analyze a data set with GEO2R. You're going to want to click expression profiling by array. And now you get a complete list of a bunch of data sets relating to schizophrenia. Now, one thing you're going to want to look at is the number of samples, as you can see here. This one's got over 5,000 samples. That's a lot of samples. But some of them will have only like 6 samples or 10. That's not enough. Generally, you want at least 20 samples in a good data set. So let's take choose a random data set here. And sometimes it takes a while to find a good data set, one that has got good samples of your disease and good control samples, but hopefully we pick a good one here with many samples. And let's take a look at this first one. No, let's take a look at this one. So when you open it, we get all different types of information. We get when it was published, the title of the study, the organism in which it was performed in, so humans, the experiment type. They should all be expression profiling by array, which is what we've done, which is what we selected for, and a summary of what's happened here. And some important things to note in your research is the type of tissue sample that your sample has been taken from. So it's blood. In this case, it's blood cells. And that matters a lot, especially with mental diseases. If it's blood samples or brain tissue, that matters a lot. And we overlook at the overall design. 91 samples of RNA isolated from peripheral blood collected. RNA is important because this is what's going to directly influence gene expression and levels of proteins transcribed and how cells differ. Going down, you have the contributor on this, and if this study has been published in existing papers, there'll be a citation here. This study has not been published to the knowledge of GEO, so there's a citation missing. Then you have your platform. This is not necessarily important for what we're going to be doing, and then you have a general collection of all of our samples, 91 samples here. All of these are all schizophrenia samples. So if you want to do research for different genes between schizophrenia and controls, maybe this isn't a great data set because there's no controls to compare it to. So if you look at this data set, we decided not great for what we want, so we'll go and try to find a different data set. let's take a look at this data set. So again, what we can do is go straight to the samples, and we see we've got controls and schizophrenia. Perfect. So the next step is, if we decided that this is what we like, it's got the right type of experiment type, these are brain samples, which is extremely beneficial for mental diseases. It's got a lot of samples, and so we decided this is what we want. We're going to analyze with GEO2R. And what we're going to do now is we're going to define our groups. So we're going to create a schizophrenia group, and we're going to create a control group. And then we're going to group each of these into their specific groups. So we're going to take all these schizophrenias, put into schizophrenia, then put all the controls into the control group. Now this can take a while, because especially with data sets with large numbers of samples, this is a pretty average sized data set. But sometimes I've used samples with over 200. I've used data sets with over 200 samples, and we saw just earlier there were ones with like 5,000 samples. And with more samples, you get more assurance in your data. So you can be very confident in the results you're seeing, most likely. And so now that we've classified everything into the correct group, we can click Analyze. And it takes a while for it to process occasionally, but when you do, you'll have a complete collection of all the information that you need to form the basis of your research. So you just wait for it to process right now. There we go. So the first thing you notice is all of these graphs. The first one I like to look at is this volcano plot. And so on this side, you've got a log2 fold change, which is a representation of differences in gene expression. Essentially what that means is, say you have a gene that's expressed 32 times more in a sample compared to another. It's log2, so it'll be log2 of 32, and that's 5. So your log2 fold change of that gene would be 5. So 2 to the 5th is 32. If it's expressed exactly 2 times as much, log 2 of 2 is 1. If it's expressed half as much, log 2 of 1/2, negative 1. On the other side is negative log 10 of the p-value, and I'm pretty sure we all know what p-values are. It's how confident you can be that, well, it's really a measure of the percent chance that a certain number is statistically significant. Now what we see here is that all these dots are black. That means that according to GEO, none of these genes, each of these dots represents a gene, are statistically significant. That means that this data set doesn't show any differences between the groups that we showed in terms of gene expression. So this is not a good data set to base our research on. This is further shown in the scatter plot, which compares levels of gene expression, and these purple dots represent the controls, and the green dots represent the schizophrenia group. And we can see they're all just strewn about everywhere. There's no order. Normally what you'd like to see is these green dots grouped together in one place and the purple dots grouped together in another place, forming a very clear boundary. This chart down here, this is for the actual individual genes. So we have the gene ID number here, then we have a gene symbol here. This is the number that you're really going to care about when it comes to our research, this gene symbol, because this is consistent across all different platforms. But as we can see here, the p-value for these genes, it's extremely, extremely high. There's very little confidence in that our results are statistically significant. Therefore, if I was doing research for myself, this is not a data set I'd be looking to use. In next videos, I'll present us with a better data set, and then I'll explain how to go from here to find specific pathways that genes are related to through KEGG pathways and StringDB. I hope you enjoyed today's modeling of how to use GEO and GEO2R in your genetic research, and watch out for the next video.